Hi, I'm going to explain Krebs cycle in this video, so please listen carefully.
The learning outcome for Krebs cycle is for you to be able to illustrate Krebs cycle. Illustrate to explain Krebs cycle. So you need to memorize all of the substrate and then able to explain what happened to the substrate along the throughout the cycle. First, what is Krebs cycle? It's a cycle that is founded by Hans Krebs, and he defined the Krebs cycle as a series of chemical reaction in aerobic respiration. So the starter or the beginning of the cycle starts with acetyl coenzyme A. Then acetyl coenzyme A will be completely degraded to carbon dioxide and water. Along the way, it will release metabolic energy that will be used to produce ATP. Another name for Krebs cycle is tricarboxylic acid cycle or citric cycle. So I will explain why they are named as tricarboxylic acid cycle or citric acid cycle, because the substrate is in the cycle. Krebs cycle is the second stage of aerobic respiration, being glycolysis as the first, Krebs cycle as the second, and it occurs in the matrix of mitochondria. So from this diagram, we have learned this step, glycolysis, that occurs in the cytoplasm. So glycolysis produces 2 ATP by substrate level phosphorylation. Okay, what happened to these two pyruvate? We are going to learn about it today. So the product from one molecule of glucose. So kita ada mula semalam dengan glycolysis, one glucose produce 2 pyruvate along with ATP and ADH plus H plus. And this is a Krebs cycle. From one molecule of glucose, Krebs cycle starts with 2 pyruvate, then produce ATP, 2 ATPs, 6 NADH, 2 FADH2, and 4 carbon dioxide. Before the Krebs cycle can begin, the pyruvate from the glycolysis will be converted to acetyl-CoA through a process named as oxidative decarboxylation. Oxidative decarboxylation occurs first. The pyruvate enters the mitochondria, and then it lost carbon dioxide. One molecule will be released from the pyruvate, during which the NAD plus as the coenzyme will act as oxidizing agent. Okay, oxidizing agent by receiving hydrogen molecule from the pyruvate. That is why it is called oxidative decarboxylation. So oxidative because this process must occur during with the presence of oxygen, and also it is coupled with the oxidation process. So NAD plus receive hydrogen molecule, then pyruvate release carbon dioxide, and then at the same time another coenzyme A will be attached to the intermediate substrate yang ada intermediate product yang ada dua carbon ni saja. So these two carbon molecule attached to a coenzyme A, thus it will name as acetyl-CoA. Acetyl-CoA will be ready for Krebs cycle. So this is a schematic diagram of Krebs cycle. Ada 8 step. Glycolysis 10 step, Krebs cycle 8 step. The unstable bond that attach acetyl group ni, this is the acetyl group to a coenzyme A, this is the coenzyme A, will break. Okay. After it is formed, the bond is considered as weak, unstable. So the unstable bond will break, will then release acetyl group to the oxaloacetate. Oxaloacetate is a 4 carbon molecule. 1, 2, 3, 4. And then we have the 2 carbon. So oxaloacetate bind with the acetyl group forming citrate. Citrate. Citrate ada 1, 2, 3, 4, 5 dan 6. Ada 6 carbon. So 4 campur, ni 4 carbon, campur 2 carbon, 6 carbon. Forming citrate. Sebab tu another name for Krebs cycle tadi, citric acid cycle. Because the first compound form during this cycle is citrate. Kenapa nama tricarboxylic acid, TCA cycle, tricarboxylic acid? Because in citrate dia ada 3 carboxyl group. Okay, jadi itu jelas kenapa. The free coenzyme A will combine with another acetyl group and then continue the Krebs cycle. Second step in Krebs cycle is isomerization. Isomerization means the citrate, the first molecule form will be isomerized into isocitrate. Citrate is converted to its isomer by removal of water molecule and then addition of another water molecule. So water molecule will first remove and then added to form bonds. Sebab dia nak rearrange kan, dia nak rearrange the atom in the molecule. So citrate is isomerized. Kita guna perkataan isomerize. Boleh ataupun converted to its isomer, isocitrate. Kalau guna isomerize, dia terus citrate is isomerized to isocitrate. The third step is oxidation and decarboxylation. Dia berbeza dengan oxidative decarboxylation tadi. Yang ini kita panggil oxidation and decarboxylation supaya awak nampak bahawa berlaku oxidation di sini. So, another name kita boleh juga sebut dia oxidative decarboxylation sebab ia berlaku decarboxylation ni penyah karbon ni kan dalam bentuk karbon dioksida. Nyah karboksil dalam bentuk karbon dioksida. So berlaku mesti dalam kehadiran oksigen. Sebab tu dia dipanggil juga oxidative decarboxylation. Tapi kalau you letak step 3 oxidation and decarboxylation pun betul. So ia menunjukkan bahawa berlaku oxidation dan kemudian decarboxylation. Ni dua benda yang berbeza. Oxidative decarboxylation means nak tunjukkan bahawa decarboxylation occurs with the presence of oxygen. Sebab ni memang aerobik kan. Manakala yang atas ni kita gunakan oxidation dan decarboxylation sebab oxidation ni refers to this process. This process. Jadi jangan anggap ia, walaupun nampak dia benda yang sama, tetapi the purpose is ni untuk NAD+. Kalau yang ni oxidative because this is an aerobic respiration. So during this process, isocitrate is oxidized. First it is oxidized and then decarboxylated. A carboxyl group is removed. So isocitrate converted to alpha ketoglutarate. The NAD+ here will accept hydrogen and reduce to NADH plus H+. Then carbon dioxide is released. In the fourth step, the alpha ketoglutarate will undergo decarboxylation first and then oxidize to succinyl-CoA. Okay, kita baca satu-satu. First, alpha ketoglutarate undergoes decarboxylation. Carbon dioxide is removed, and then the resulting compound. Kita guna perkataan the resulting compound sebab tak ada nama spesifik pada masa ni. The resulting compound is oxidized by NAD+. So NAD+ reduced to NADH plus H+, and the remaining molecule is attached to coenzyme A to form succinyl coenzyme A, succinyl CoA. Okay, ni tak sebut by an unstable bond pun tak apa. Yang penting you tahu bahawa the attachment of coenzyme A to any molecule is an unstable bond. Step 5. Dalam Krebs cycle hanya ada satu step saja yang hanya ada sekali saja substrate level phosphorylation, iaitu step kelima. Langkah kelima ini, succinyl CoA will lose coenzyme A first to form succinate. Okay, dia akan hilang dulu. Succinyl CoA ni will lose its coenzyme A. And then during this process, phosphate inorganic here, phosphate inorganic will be transferred to GDP. The phosphate inorganic ni dia macam attach sekejap dengan proses ni sebab apa? Semasa succinyl CoA, during the release of coenzyme A from succinyl CoA, it releases energy, right? Energy is released. So this energy is used to bind phosphate inorganic towards GDP. So we can say that GDP receive phosphate to form GTP. Okay. So GTP will transfer the phosphate kita dah cakap the phosphate, tak boleh cakap phosphate inorganic lagi. So GTP then will transfer the phosphate to ADP and form ATP through substrate level phosphorylation. So the succinate is formed. Ada soalan? Next is conversion of succinate to fumarate through oxidation process. So the oxidizing agent here is the FAD. Remember, FAD is also a cofactor. FAD is a cofactor, but it's not a coenzyme. It is a prosthetic group. It is an example of prosthetic group. So FAD will oxidize succinate to fumarate by receiving hydrogen molecule. So FAD will be reduced to FADH2. Next, fumarate will be converted to malate through by the addition of water. With the addition of water, the molecules in the fumarate will be rearranged and then converted to malate. So very simple, fumarate converted to malate with the addition of water. The next process is oxidation and regeneration of oxaloacetate. Okay, so malate will be oxidized. Malate will be oxidized to oxaloacetate by transferring hydrogen atoms to NAD+. So NAD+ is reduced to NADH plus H+, then oxaloacetate is ready to combine or start another Krebs cycle with acetyl-CoA. Ada soalan? Tak ada jadi kat sini one turn. Maksud one turn ni satu pyruvate. Since we produce two pyruvate from glycolysis, so we should have two pyruvate. So we should have two turns. Okay, two turns. Two turns or one turn of Krebs cycle will produce one ATP. Okay, here 3 NADH plus H plus 3 NADH plus 3H H plus, here 3 NADH, 1 FADH2 and 2 carbon dioxide. Sorry, ini bukan Krebs cycle. 2 carbon dioxide, okay, 1. Jadi kita tengok pada dia punya diagram. So for
Ini
one
madam.
yeah.
Ini madam, yang one turn tadi madam cakap apa madam?
One turn maksudnya satu pyruvate. So kita ada 2 pyruvate, dia patutnya ada 2 turns lah.
Okay, thank you madam.
Okay. So ni 1 molecule of glucose, meaning we should have 2 turns of Krebs cycle. Okay. Jadi Krebs cycle kalau lengkap satu glukos, maka sepatutnya kita ada 2 ATP, 6 ADDH, 2 FADH2 dengan 4 carbon dioxide, 4 carbon dioxide. Kita tengok ikut gambar rajah yang penuh kat sini. Let's identify where ATP is produced. First ATP here. 1 NADH patutnya ada 3 tempat, iaitu step 3, step 4 dan step last 8. FADH2 ada 1 saja. Right. Carbon dioxide di sini ada 2. Okay. So the net produce, kalau kita ambil Krebs cycle tadi 6, 2, 2, 4. Manakala bagi Lee reaction kita tak boleh abaikan. Okay, sebabnya nanti NADH akan diambil kira untuk menghasilkan ATP dalam oxidative phosphorylation. So through link reaction, we also release another 2 carbon dioxide. Kenapa 2? Sebab kita ada 2 pyruvate. Okay. Then 2 acetyl-CoA and 2 NADH. Jadi this one go to oxidative phosphorylation. Ini release from cell. Release lah, kita akan exhale. Acetyl-CoA will go to Krebs cycle. Faham? The total ATP produced by substrate level phosphorylation melalui Krebs cycle saja. Link reaction tak ada substrate level phosphorylation. Jadi Krebs cycle produce 2 ATP through substrate level phosphorylation. Sekarang ni bila kita belajar, kita dah ada 4 ATP yang dihasilkan melalui fosforilasi peringkat substrat. Okay, now let's focus on NADH and FADH2. NADH and FADH2. So NADH and FADH2 function as electron carrier to carry the electron towards ataupun to oxidative phosphorylation. Okay. Ini mnemonic untuk TCA cycle. Can I keep some succinate for myself? Jadi ini mungkin membantu awak untuk mengingat nama-nama substrat dalam Krebs cycle. Oh, can I keep some succinate for myself? Oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate.